First and foremost, we are going to study recombinant DNA technique or technology. This is also known as genetic engineering because we are going to do some modification in the DNA by combining DNA of two different sources in a suitable vector, thereby we altering the gene expression. So for this we need to have a DNA and the vector. Then we are combining these two. This is called hybrid DNA or chimeric DNA. Please remember, chimeric DNA is a recombinant DNA containing genes of interest from two different species. So, what are the steps of DNA recombinant technique? This is your SLO. The first and foremost is we need to fragment the DNA. This usually from human DNA, so which contains a gene of interest. We are going to cut this particular gene by a special molecular scissors. They are called restriction enzymes, are also known as endonuclease or restriction endonuclease enzymes. Once we fragment intended DNA, then we need to separate from other fragments. That means maybe we have entire genome, but we cut the specific intended DNA. We need to isolate only these particular fragments. For this is the second step. We can use electrophoresis or HPLC. The third step, once we fragmented and isolated, we need to insert this small portion of DNA which is has a intended gene or nitrogenous bases or genetic codes. We need to insert into a suitable vector. Okay, so that is the DNA of different species or sources. So this particular process is called chimeric or hybrid DNA formation. So these two DNA of different sources are joined by DNA ligase. That is the fourth step. We already learnt in DNA replication what is the functions of DNA ligase. Okay, we'll see one by one now. So as I mentioned earlier, the first step is fragmentation of DNA. Okay, so how to cut double stranded DNA? For this, we are making use of prokaryotic enzymes usually found in bacteria. So these bacteria have specific enzymes. They can cleave both double-stranded DNA. They are known as restriction enzymes or restriction endonucleases. So in your mind, you may have doubt if these prokaryotes or bacteria process these enzymes, if they are having the ability to cut double-stranded DNA, then why don't they cleave bacterial DNA itself? Here, bacterial DNA are protected from restriction enzymes by methylation. Okay, so these restriction endonucleases cannot cleave methylated bases. And please remember the specificity of these restriction endonucleases are not random. Rather, it is specific. Each restriction enzyme have its own specificity. What I mean to say, double stranded DNA, the sequences are palindromic in nature. So palindromic means if you see here I have a piece of double stranded DNA. If you read the code from 5 prime to 3 prime, you can see GAATTC. The same sequence, if you read from the bottom strand 5 prime to 3 prime, same sequence, you can see GAATTC. So these sequences are called palindromic sequence. Okay, palindromic sequence. Most of the restriction endonuclease enzyme, they can cleave only their specific sequence and also they should be in a palindromic manner. Otherwise, they cannot cut the both the end. So the name of these enzymes, there are many enzymes. The first and foremost restriction enzyme isolated from the bacteria, which is isolated from E. coli or Escherichia coli. This we call it as ECO R1. Okay. So like that, HAE3, that is isolated from Haemophilus aegyptus. So you should know everything about restriction endonuclease enzyme. They are isolated from bacteria. They recognize specific base sequence which are palindromic in nature, and they also cleave both the strand. Okay. And their name according to the bacteria they are isolated. Now these enzyme they can cut double stranded DNA. The manner in which they cleave double-stranded DNA produce two types of ends. Okay, one is cohesive end or sticky ends. Another one is blunt ends. Okay, so those enzymes which produce cohesive ends or sticky ends, they cut so that there will be unpaired strand. Okay, you can see here these TTAA sequence are not paired. Even in another cutting fragment, you can see here AATT is not, it is unpaired. Okay, they produce staggered cut on the two strands, producing unpaired strands. So this is called cohesive ends or sticky ends. Some another restriction enzymes, okay, small or HPAL, okay, they produce blunt ends. That means they cut the double stranded DNA without producing any unpaired bases. Okay, so please remember the result of restriction endonucleases can be cohesive ends or sticky ends or blunt ends. Okay. So once we fragmented, so what is the first step? Fragmentation. What is required? Restriction endonuclease or restriction enzymes. What is the source of restriction endonuclease? It is bacteria or prokaryotes. So what is the specificity? They must have palindromic sequence. And there are two types of end, cohesive end and blunt end. Okay. The second step is isolation of these fragmented DNA which have a specific genes. Okay, code for specific genes. So this, as I mentioned earlier, the separation technique either by electrophoresis or HPLC. HPLC means high performance liquid chromatography. Okay. So now we'll move on to the third step, which is we have fragmented, we have isolated. Now this isolated human DNA sequence, okay, we need to insert into a specific vector, produce hybrid DNA molecule or chimeric DNA molecule. These vectors could be bacterial plasmids. So bacterial plasmids means bacteria have their own chromosomal DNA. From this, they also have small circular double-stranded DNA, which we call plasmids. Their main function is to exhibit antibiotic resistance property of the bacteria. Okay, that is bacteria plasmids. They are usually present in the bacteria. Another one is bacteriophages. Okay, you have heard macrophage. Okay, phagocyte. Phages means eating, bacteria eating. That means the virus which can kill bacteria, which can infect bacteria, they are called bacteriophages. They are also very good vectors where we can insert the fragmented DNA. And last one, these two are naturally occurring. This is from the bacteria, this is from the virus. The last one, cosmids are It is hybrid or it is combination of bacterial plasmids and bacteriophages. That means we can produce these cosmids. Okay. It is artificial vector. The fourth step is once we inserted intended DNA into a suitable vector, then we need to join them by DNA ligase, which you already learnt in DNA replication. Okay. Now, now we need to cloning of this DNA or amplify this DNA. Okay. Once we make chimeric or hybrid DNA, so we can insert into a suitable host cell and it can modify or amplify intended DNA. This is called amplification of recombinant DNA. So whatever DNA we produce, we can insert into a host cell so that it will produce apart from host DNA, this DNA, recombinant DNA also multiplied, divided and multiplied, and this is called cloning. Then in vivo, inside the body or inside the host, we call cloning. And similar technique we can use in PCR, it is in vitro or outside. There also we are amplifying, that is PCR. Okay. So this is with respect to recombinant DNA technique steps. Now we need to see what are the application, what are the uses of this genetic engineering technique or recombinant DNA technique. First, we need to understand the molecular basis of some of the disease, or in other words, mutation. To study mutation like thalassemia, cystic fibrosis, or any other mutation, so we can use this recombinant DNA technique to study, to make a diagnosis, or to study or to understand molecular basis of some of the inherited disorders. We can use this technique. Similarly, in the lab, we can produce insulin. Okay. For diabetic patient, we need to inject insulin, that we can produce this insulin and factor 8 for hemophilia patient. So we can produce these two proteins. Apart from these two proteins, many other proteins can be produced by using recombinant DNA technique artificially in the lab. Okay. And also we can produce many vaccines. First vaccine produced by recombinant DNA technique is hepatitis B virus vaccine. So now you understood that there are many applications or clinical importance of recombinant DNA technique. So not only these molecular basis or diagnosis, production of protein, vaccines, even recombinant DNA technique are utilized in gene therapy. Currently, it is at experimental level. In forensic medicine or medical legal cases, to identify criminals, we can use DNA fingerprint that uses recombinant DNA technique. This is about recombinant DNA steps and uses.